How's everyone doing? You know, it's so interesting to be at this conference and talking about biohacking and plants and plant medicine, botanical medicine. Super exciting topic. And sometimes we can get into our heads and we can start creating these memes about how botanical medicine is natural and thus is good. You know, we are in a constant battle. We're at war with plants, and plants are trying to attack us, and we can actually make medicine out of these attack mechanisms. So, there you go. So what are we going to talk about here? We're going to talk about how evolutionary adaptations of plants can be beneficial to humans. We're going to talk about the potential of medicine and the things that we can do with these plants and how we can use an evolutionary approach and evolutionary medicine to actually create really cool therapeutic effects. We're going to identify different ways that we can predict treatment protocols for these plants. And we're gonna recognize the importance of being judicious with this medicine so we don't fall into the same problems that we've fallen with our pharmaceuticals. So I want to tell you a little story. In Venezuela right now, they are suffering the worst inflation that they've ever suffered in the past. They are literally carrying wheelbarrows of money to the store to buy their groceries, and they stopped counting money. Now they're just weighing it. And there is not a lot of food to be bought. So people started digging around, and families started not recognizing yucca, and they were digging actually bitter yucca. And it's called bitter yucca because it has high levels of cyanide. Now, yucca can be, pounded to a flour, and you can make different types of breads with it. But if you were unfortunate and you found bitter yucca instead, and you made this bread, you basically die. And this is the mechanism that yucca is using to fight you from eating it. And just imagine how sad it is that we think of, this plant, it's natural, it's gonna be good for me. But in reality, that plant, just like you, is just trying to survive. Now, are these plants, trying to attack us, or, can we have a symbiotic relationship? This right here is the tobacco plant. And this is a really interesting plant. I love alkaloids. I love nicotine, caffeine, and all of those things that can attack us, in our brain. And they bind to certain receptors, and they make us feel awesome. They make us feel excited. Now, all of these plants are developing these alkaloids as a way to defend themselves from insects. All of these alkaloids, like nicotine and caffeine, are actually insecticides. And these plants are so good at making these alkaloids that eventually the plant was making so many of these alkaloids that pollinators stopped pollinating the plants. So, this in evolutionary terms, if you don't have someone pollinating you, then you're going to have to develop a different strategy for survival. And in fact, the tobacco plant actually started self-pollinating. But if you think about it, and this is really exciting, we can use these plants for our benefit, or so we think, because who is controlling who? Are these plants developing these extracts in order to be pampered so we can continue cultivating them? Or are we cultivating them for our benefit? And I'm going to give you a brief example. We have, for example, the coffee plant. And the coffee plant, it makes this alkaloid, caffeine, in a very specific way. It creates this big molecule, and it starts chopping it up until you have this awesome alkaloid caffeine. The chocolate plant actually does it in reverse. It puts together all of these different compounds in order to make this alkaloid. both of them insecticides, both of them are they kind of make us excited and they make us crave these plants. So these are different ways that these plants can create adaptations in order to preserve themselves, and we can gather those adaptations and benefit from them. Now, I got interested in botanical medicine when I was in school because there's a lot of traditional education and traditional information telling us that these plants are really useful. And I particularly became very interested in carnivorous plants, or plants like, for example, this right here is Sarcenia purpurier. And Sarcenia purpurier is a native plant of the northeast, and because it grows in ponds and in swampland, and that land has very little nitrogen content, this plant evolved to consume organisms to satisfy its protein requirements. Like, how cool is this? This plant was incorporating protein into their diet before the ketogenic guys told us that we needed to do that. So these guys are pretty advanced. In fact, Darwin said that carnivorous plants are the most wonderful plants in the world. And you know, when I was in school, someone raised their hand and they said that, oh, you know, these natural plants are really cool because since they're natural, then they are completely benign. And I was scratching my head and I was like, well, is there any truth to this? If a plant has an effect, then it must have side effects. But where to start? And we found this paper from the BMJ that talked about the Mcmac Indians. Now, it was very interesting because in this population of Indians, there was an outbreak of the pox virus. And there's not really a lot of different things that can fight the pox virus. But the BMJ stated that amongst the people treated with this plant in this location, none of them died from the pox virus. Now, I have access to a virology lab, and I wanted to see, well, what's going on with this plant, and how can this plant be able to fight this virus? Well, we tested it in vitro, and we found that if you do use an extract of this carnivorous plant against cells that have been infected with this virus, you have really cool results. So this right here is just healthy cells, and after you infect them with the pox virus, you can see how they go from squamous and they start to apoptose and they blow up and they die. If you use the extract of Sarcenia purpurier, well, the cells survive. So these are cells that were infected with the virus, and because they were treated with this antiviral extract, they survive. So what's happening in this to these cells? Well, we were able to publish our discovery, and we have a series of explanatory research papers saying what is happening with this extracts. And I'm gonna make it a little bit easier for you to understand. You know, so you have plants, and plants are being visited by different animals or different bugs. And these bugs and animals act like vectors. And when these vectors land on these plants, then you start exposing these plants to different viruses and bacteria. And if a plant has to eat these viruses and bacteria, it has to develop endogenous substances in order to fight these infections. And you know, believe it or not, the immune system of plants is very, very similar to that of humans. So is this only present in carnivorous plants? So we decided to do something kind of neat. We actually investigated all the different clades that eventually developed to being carnivorous, and we found common ancestors of plants that were carnivorous or not carnivorous, and we decided to test them against these viruses. And what we found out was that in this close proximity, we tested those different plants, and only the plants that were carnivorous, that developed the different proteases and the requirement of incorporating amino acids through digestion, only those had the antiviral activity. And that was really, really exciting, because now we found the nexus between evolutionary theory and traditional medicine. So imagine if we could use all this traditional knowledge to predict how different plants would act against different types of viruses and bacteria. So we wanted to put real evidence behind these discoveries. And we tried these extracts against all sorts of viruses, like filoviruses and DNA viruses and RNA viruses. And it turns out the Sarcenia Purpurier and the carnivorous plants extracts don't work against things like rotavirus, rabies, or coronavirus. But it was very effective against DNA viruses. Of the DNA viruses, you have things like smallpox, like that original paper we published, and then herpes virus that causes things like cold sores, or HSV2 that causes shingles, and then papillomavirus, which causes warts and other things that can become precancers. So if we were able to discover that, okay, this carnivorous plant is really good against viruses, and these viruses happen to be of the DNA family, okay? Without testing, could we predict what other viruses this plant can be useful against? And a very well-known DNA virus is the HIV virus. So we decided to see if by just predicting this mechanism of action, if we would be able to destroy a different virus that we never tested against. And sure enough, we were able to reduce viral load just by prediction. So imagine a world where if we can identify the actual mechanism of action of the plant, then we can go back and reverse engineer and apply it at different stages or towards different types of conditions. So as you can see right here, you have the ethanolic, just an ethanol carrier, so there's no plant extract, or the plant extract. And at 0, you have not a lot of suppression of the virus. And as you increase the concentration of the plant extract, you see complete suppression of the virus in comparison to the carrier. So what's happening within Sarcenia Purpurier that is effective against this type of viruses? Well, these types of viruses replicate using a pathway called CDK7. And CDK7 is what phosphorylates this virus and eventually shuts down P53. So what we see is that Sarcenia Purpurier and all of these carnivorous cousins are able to block that CDK7 pathway and allowing P53 to do its thing and send the cell into apoptosis. Now, one of the problems with viruses that use CDK7 is that they can convert regular cells into cancer cells. So again, with our predictive mechanisms, could we predict that these plant extracts were going to be effective against transformed cells? These are cells that have already been invaded by the virus. All of their machinery was overtaken, and now this cell is acting on its own and replicating without anyone telling them to stop it. So this is HPV cells, and you can see uninfected cells that have been already transformed. Then if you just let them continue replicating, they eventually stop reproducing and they apoptose. If you use those uninfected cells plus Sarcenia, you rescue the cells. So it can break down the mechanisms where the virus is taking over those cells and allowing them to continue replicating until they die. Now, this is a very cool experiment right here, because these are transformed cells, transformed cervical cells that are immortal. These cells continue to replicate just like cancer. And this might be a little confusing, but this is the first day post-treatment, so this is day 0. This is how you inoculate the Petri dish. You put a couple of cells, and by day 6, the whole Petri dish is completely covered with cancerous cells, meaning that they just continue replicating. By adding the extract of Sarcenia purpurier, surgically with precision, the plant extract starts eliminating only those cells that were transformed, because it's acting on the same mechanism of action as the virus and preventing the phosphorylation of CDK7. And you can see this is the growth of cells. So this is the vehicle or without treatment. And if you don't treat the cells by day 7, it has completely covered the whole plate. But if you use a Sarcenia purpurier extract, you see that growth is completely eliminated. Now, that's super exciting, because we can go back and, we have a native population that thought, oh, maybe we should use this plant in order to treat this crazy virus we've never seen. So then we can use that traditional knowledge coupled with hard science to identify the mechanism of how is that plant working on that virus. And then using evolutionary thinking, we can predict what applications we can have for each one of these extractions. So now imagine if we could do, we can do all of this, and we can have this thought experiments, and we can put them in vitro dish, in an in vitro medium, and then we can make them work, and it's very exciting, and I get to present here. And then someone always raises their hand and says, well, that's in vitro. The cool thing about botanical medicine is that we traditionally can use herbs yesterday. We don't have to go through a bunch of approval, and we don't have to, do a lot of experimentation, because these plants have been proven safe for thousands and thousands of years. So what we can do is we can take these concepts and do case studies, N equals 1, with people that have this type of cancers. So far we have done 3 different case studies where people with genital warts, HPV, HSV2, and actinic keratosis, and those all of those types of cancers work on that CDK pathway. And because it's only plants, we have been able to test them, and we have seen complete eradication of those tumor growths. And that's, from that traditional knowledge all the way to hardcore science. Now, what other things are working on that CDK pathway? Well, things like neuroblastoma, prostate cancer, Epstein-Barr, lymphomas. So all of these different applications can be deduced by knowing the exact mechanism of action of the plant. Now, we have to be super careful, because if we continue abusing this medicine, or if we don't understand what the actual mechanism of action of these plants actually is, and we just continue using them without knowing, then we can create problems of viral resistance or cancer resistance. And that's why it's so important to educate ourselves before we try to use all this plant medicine and on ourselves, or try to do it without the supervision or without the knowledge. These plants are pretty safe. They're well tolerated, but we need to be very careful. The number one concern by the WHO by 2050 that it's not going to be heart disease, it's not cancer, it's not autism, it's not Alzheimer's, it's bacterial resistance. We have thousands and thousands of plants right outside our backyard that could be the answer to fight bacterial and viral resistance. So we need to be very judicious. We need to understand mechanisms of action, and we need to learn how to properly extract this plant material in order to make it the most effective. Now, not all plants are antibacterial or antiviral, and for this reason, all of you biohackers, I've put together the top supplements for your natural medicine cabinet. It's a free PDF with double-blind, placebo-controlled trials of different things that you can use on your medicine cabinet if you want to use something other than the pharmacologic agents. Pretty cool things, like for a cough, you can use a combination of coffee and honey, and that's more effective than taking dexamethasone for a sore throat, or using ginger to help you with nausea. So it's really cool resource. Yeah, it has a cool table of contents where you can rapidly access different remedies that you can take, and they're very safe, and you're not going to get in trouble by creating more bacterial or viral resistance. I do have a podcast, and I do talk about all this crazy science stuff. And if you guys have any questions, I'm opening the floor for questions. Can you use the mic for us?
What's your podcast called?
Oh, 30/30 Health.
Go my tippy toes. I was wondering, it's kind of completely off topic, but do you know if once you're taking antibiotics, if you should continue the course, or it's better to stop when you feel better? Because I feel like it makes more sense to continue the course. You're not leaving stuff that's probably resistant. But I've also heard that you should just stop as soon as you feel better.
So, antibiotics, very, very controversial topic. Antibiotics do affect your biome, but there are things that we can do in order to prevent that biome damage. They have been thoroughly studied, and they have been, standardized for efficacy. And when you are prescribed a course of antibiotics, you should damn well take the whole thing if you don't want to create resistance to bacteria. The problem is that a lot of these herbs that we use as antimicrobials, things like sage, things like rosemary, grapefruit seed extract, all of that, they can create, they have very similar mechanisms of action as antibiotics. And if you're not under the care of a physician or someone that understands how to use these herbs, and you use them at the correct timing and for the correct reasons, you can create bacterial resistance with botanicals. I actually have a paper on the Journal of Evolution and Health on that same topic, how we were able to create bacteria that was resistant to vancomycin using herbs. They were never touched by any antibiotic. We were just using herbs, and that's a problem, because in my practice, sometimes I'll prescribe an antibiotic, and people are like, "Oh no, I don't want to take an antibiotic," but they are willing to grab a bottle of water and put a little bit of essential oil and drink it all day long. In essence, taking a diluted antibiotic all day long, and that scares the hell out of me. So, if you have an infection that was identified, via a stool analysis or a swab, and there is an antibiotic that has been standardized and has been well studied, take the antibiotic and save herbs for things that are a little bit more nebulous and not as well characterized. Yeah, you're welcome.